Today we're going to be looking at DNA and kind of an intro of who discovered it, its basic shape. Also, you'll be looking at what base pairings go with which. In other words, if one letter is on one strand of the DNA, what's on the other side of it. Also, we're going to be introduced briefly to what transcription is. Let's take a look. Remember, DNA is a nucleic acid that is located in all cells, contains all information about the organism. Remember, DNA is a complex molecule that stays within the cells and dictates what the cell needs to do. It is the site of all the information for the cell, and it's stored in a nice compact molecule. It's what makes you, you, as well as determines certain traits that you will have throughout time. Who discovered DNA? Watson and Crick discovered the structure of DNA. Watson and Crick were two scientists at Oxford University who, with the help of radio technology, were able to discover the shape of DNA in its double helix form. From there, they were able to pinpoint and understand the base pairings of DNA and how this complex molecule works. Structure of DNA: double-stranded, has one strand going one direction and one going the other way. The double-strandedness of DNA allows it to contain even more information than a single strand. This way, not only is one side able to be read for specific information, but the other side also can contain complementary information or its own set. They are complementary strands. We call this the 5' to 3', with one end of the DNA being the 5' end and the strand on the other side being the 3'. Note at the top of each of these strands is a 5' end and a 3' end. This indicates which way the DNA is read, as it is always read from one 5' end to the 3' end. Think of it this way: the DNA is count backwards, from the beginning reading at the 5' and it reads down to the 3' end. Matching base pairs. DNA has 4 bases: A, C, T, and G. The bases always match up in a set order. A bonds with T and G bonds with C. If we observe the DNA and the bases within the center of it, we notice that it has the complementary base pairs, with A, adenine, binding with T, thymine, and G, guanine, bonding with C, cytosine. These pairs are always the same and will never change. This way, you know if you see a T on one side of the DNA, on the other strand must be an A. Likewise, if you see a C on one side, a G has to be on the other side. DNA strands are written like this. Notice there's the 5' and 3' at the beginning, followed by the complementary base pairs on either side. Has the ability to unzip from other strands and be made into protein. This process is called transcription. Since DNA is so large and cannot move, transcription is the process where it will send out individualized messengers to the rest of the cell to indicate what to do for survival. Differences between DNA and RNA. DNA is different from RNA in several ways. DNA is double-stranded, and RNA is single-stranded. DNA's structure allows it to be double-stranded. This means that it can contain more information. RNA's structure is single-stranded, as its main purpose is to transmit information. DNA equals deoxyribonucleic acid. RNA stands for ribonucleic acid. RNA has a ribose sugar in its backbone. If you observe the pictures as following, you notice that DNA has a deoxyribose sugar and RNA has a ribose sugar. This allows it to have different structures, leading it to perform different functions. DNA is as a storage, and RNA is more of a transmitter. DNA has a nucleotide of thymine, and RNA has a nucleotide of uracil instead. As we've seen before, RNA has a U. It's similar to thymine, it just has a slightly different structure. DNA stays in the nucleus of complex cells. RNA is found all over the cell. In complex cells, because they're very large, it pays to have the DNA in a site location. This is the nucleus. However, because information needs to be spread to other parts of the cell, RNA is used to transmit that information.